Hello students, welcome to another tutorial video from this Agri Education. And today all of you the students, I think this chapter is very important. This is Uni-II, topic number 4. Subject is photosynthesis. This particular topic has been divided into two different main section. They are light reaction and dark reaction. I will discuss each and everything related to phosphorylation under light reaction, and another one is dark reaction, that is also again divided into C3 and C4 cycles. And C3 and C4 cycle, that is some difference between C3 and C4 cycles. All the things related to electron transport chains will be discussed here. So be concentrated. Within a very short time I will cover all the topics. So at first, definition as it has been modified from the from your books, and I have made this definition to easy understanding. It is a biochemical process by which photosynthetic organisms convert light energy into chemical energy, NADPH and ATP, and use these chemicals to drive CO2 fixation and reduction to produce sugar. Very easy in the sense chemical energy. First step is light reaction. Light reaction, what it is going on in the light reaction? They are producing NADPH and ATP. These are the both energy producing molecules. So these particular compounds are in the first steps has been synthesized. In the second step, that has been utilized by with the help of CO2, and here carbon assimilation or carbon fixation is going on, where CO2 has been used in the form of carbohydrates, where you can see the carbohydrates, where the carbohydrates carbon molecule is coming from CO2, and here ATP is required for energy consumptions. So reduction, it is again reduction process. That's why this is the dark reaction. So overall definition has been completed by light and dark, in combination of light and dark reaction. So overall equations has been given earlier. CO2 plus 4H2O with the help of light and chlorophyll. Chlorophyll is the center of this particular photosynthesis, where we can see CH2 whole bracket plus 3H2O plus O2. So you can say, sir, how it is possible CH2 whole bracket you have given here? So this is the preliminary formula from which the glucose molecule has been prepared. Okay, you have to be understand this thing. So site, what are the sites of photosynthesis? The most active photosynthetic tissues of higher plants is mesophyll. So you have to be remember mesophyll cells are very sensitive, and where the particular site of photosynthesis has been happened. It contains a specialized light absorbing green pigments that is called chlorophyll. So chlorophyll has two different types. You have to be just remember only chlorophyll A and chlorophyll B. Most of the cases, in case of C3 and C4, chlorophyll A is taking part in 90% cases, and chlorophyll some parts of whole photosynthetic pathway. Chlorophyll B is also taking. The region of the chloroplasts that inside the inner membranes and surrounds the thylakoid membrane is known as stroma. It contains the enzymes that catalyze carbon fixation and other biosynthetic pathways. The thylakoid membranes. So how you can differentiate between thylakoid membranes, grana, stroma and chloroplast? So I will see the details in the later slides in this video lectures. So what are the each and every sentence in this particular topic is very important. You have to be careful about this particular underlined sentence. Okay. So next one is light as a biological agent. Why light is used, or why light is important for the synthesis of carbohydrates. So absorption spectra are the graphical representation of quantity of absorption of the different wavelength of the light by any pigment of the chemicals. So students, it has to be remembered that plants only used mostly the red color for their energy consumption in the form of absorption spectra. They actually taking out the electron in pair, so whole photolysis events as well as electron transport chain has been carried out to the passing of this particular electron, excitation of this electron. All the things are under this category. So green rays of the light are absorbed least. So just see here, green rays are the least amount and red will be the highest. Transmitted through or reflected by the leaves in majority. Leaves are green, that's why. Because leaves are green, that's why they are taking the least amount of the green rays from the visuals colors. On the other hand, red and blue wavelengths of the light are absorbed, uh, light are absorbed maximum amount. Only wavelength of visible light range is suitable for the photosynthesis. So another question can come, whether photosynthesis can be taken place under tube lights or white light conditions. Yes, students, under night conditions is sufficient amount of wavelengths of the lights will come from artificial lights. The plant can photosynthesize if they required. Okay. In the context of chloroplast, light energy is converted into the chemical energy by two different functional units called photosystem 1 and photosystem 2. So next one is photosystem 1. Photosystem 1 absorbs far red lights of wavelengths greater than 680 nanometer. That is basically 700. So photosystem 1 and photosystem 2 absorbs the red light which is about 680 nanometer wavelength. The ratio of PS2 and PS1 will be 1.5:1. So the role of PS2 is much more than PS1 because ratio is more. So utilization of non-cyclic phosphorylations will be carried out mostly by PS2. So immersion effect is what? Immersion effect is the enhancement of quantum yield when two different wavelength, blue and red, applied simultaneously. If some plants will get a sufficient environments where blue and red wavelengths will be highest, so photosynthetic activity will be increased from the normal conditions rather than the normal conditions. So this can be achieved to production of higher amount of carbohydrates, and plants photosynthetic activity will be increases. So photosynthetic pigments, as you know, chlorophyll A and chlorophyll B. Other associated pigments are there, carotenoids, xanthophylls presents in the plants. But in case of photosynthetic actively participating bacteria, some bacteria are capable of photosynthesis. They have used phoicobilin in the place of accessory pigments for photosynthesis activity. Capturing the solar energy, this is also important. Photon absorbed by antenna complex and electron processed through the chlorophyll to chlorophyll by excitation transfer reaction, and finally channel to the reaction center and thereby thylakoid membrane. So a common xanthophyle of leaves is lutein. This is also questions coming. And a common xanthophyle of algae is fucoxanthin. The questions are coming. Plastids, what are the plastids? Different part is consists basically chloroplast. It is one type of plastid having green pigments. Chlorophyll only content with the green pigment. Chlorophyll is of two types, chlorophyll A and chlorophyll B, and xanthophyle also under chloroplast category. Chromoplast is basically colored pigments what plants produced in the different parts of the tissues. You can see different colors, gray, red, maybe yellow, maybe white, maybe blue colors. Different types of fruit colors it has been developed. So this is basically due to chromoplast. Carotenoids, it is inducing the pollination and fertilization and dispersal of the seeds by animal body for the purpose of most of the flowers. You can see that's why the color is carotenoid means orange color, that is very very attractive color. So next one, leucoplast. Leucoplast basically used for white color. Under this different types of leucoplast are amyloplast, carbohydrate synthesis responsible for this, elioplast, fat synthesis, and proteinoplast for protein synthesis. Questions are coming. Chloroplast, the photosynthetic reaction center. We can see this is the structure of plastids. We can see here chloroplasts has been divided. Chloroplast has this particular structure, but the chloroplast this is the elioplast, this is the etyloplast, and chromoplast, this is the amyloplast. So chloroplast has this type of thylakoid structure, thylakoid membrane like structure. One by one thylakoid membranes are put in a stack like structure, stuck together, and this is, you can see here, fluid inside the membrane in the stroma where tiny membrane sac floating on its thylakoid membrane. So what is this, the thylakoid membrane? This is the thylakoid membranes where the fluids are situated. Thylakoids are stacked in group called granum. So you can see this is the whole part we'll call granum. One by one granums are stacked together to make a thylakoid membrane, where a number of granum present in stroma is called grana. When 1, 2, 3, here you can see 1, 2, 3. 3 different granum are stuck together and form that as a grana. Grana interconnected by stromal lamellae. You can see one, this is the grana, and another grana that is connected by stromal lamellae. Very easy. Or intergrana is also called. Next one. Thus, thylakoid is the site of light absorption and ATP synthesis. This is the whole thylakoid membrane that covers the granum and stroma. So, what is the difference between stroma and their functions during the photosynthesis will be taken care. Photosynthesis occurs in two different phases: light reaction and dark reaction. Why it is light reaction? Because light dependent reaction and light independent reaction. You just can see light dependent reactions we can write down here, and light independent reactions. So because in this case one question can come that whether during the dark reactions whether light dependent reaction is possible. Yes, what are the light reactions are going on, that is also can be possible during the dark conditions, because it is light independent. It can be possible under the light or without light. Both can be possible in case of light independent reactions. So light reaction, what it is happening? It uses the light energy to synthesize NADPH and ATP at thylakoid membrane. The site is thylakoid membrane. The absorption of the light energy by the chlorophyll molecule and its conversion into the chemical energy and storage of ATP from breakdown of ADP and PI is called photosynthetic phosphorylations. What it is mean? That means in this process, the particular light energy will be entered into the PS system, photosynthetic system, where one by one electron has been absorbed in the form of paired structure, and this particular photosynthetic system will be excited to a particular level that is basically called photosynthetic electron acceptor site. Okay. After getting excited, it will start to reduce by transferring it through a series of photosynthetic acceptor molecules or carrier molecules, you can say. Okay. So first one, in case of non-cyclic photophosphorylations, it will come. So cyclic photophosphorylation, what we can see? Cyclic photosynthesis is associated with cyclic transport of electron from photosystem 1 via plastoquinone, cytochrome BF, B6 complex and plastocyanin and photosystem 1. In this process, large number of AT molecules are produced and this energy is used in dark reactions. You can see whatever the ADPs are produced in the light reaction will be utilized into the dark reaction for the synthesis of carbohydrates. During ETC, electron transport chain, PS1 returns to its ground state by releasing electron, leads to a sequential electron reduction and thus causing proton gradient. So what will be the happening? Now you can see in this picture I am drawing here. Suppose this is non-cyclic photophosphorylation. What it is happening? At first, this particular electron will be then transferred to the lower amount of different electron acceptors and others carrier. In case of non-cyclic photophosphorylation, at first it will enter into the plastoquinone. After that, it will transfer to again to cytochrome, and after that you can see here plastocyanin, another electron acceptors, and after that it will transfer to the electron will be transferred to PS1, photosystem 1. The absorption spectra is, in this case, is 700. Okay. So in this way, non-cyclic. So it is the one way of process. There is no cyclic reaction is occurring. That's why this is called non-cyclic photophosphorylation. Photophosphorylation, why it is means during the time of this conversion, ADP adenosine diphosphate attached with phosphate molecule and produce ATP. So phosphorylation take place. That's why this is called phosphorylation, photophosphorylation under the photosynthesis, non-cyclic, because this is the linear chain cyclic linear chain reactions. So after entering into the PS1, it will transform this particular PS1 cyclic reactions, where it will again transfer to photosynthetic electron acceptor site, and transferring these sites, this particular acceptor will be excited. To minimize this, it is again transfer the electron to ferredoxin. This is the different here. There it is direct plastoquinone in case of non-cyclic, here cyclic in case of cyclic. This is ferredoxin. After ferredoxin, it has entered into the plastoquinone, and after again in the simple way, this is the events similarly happening here, and again plastocyanin, cytochromes, and after that plastocyanin, and this plastocyanin will transfer this electron to NADP. This is already within the cell, and after getting this electron, it will be reduced to NADPH. So where from it will take this H plus ion. Now you can see in the fast stage, when non-cyclic photophosphorylation is occurring, in this case basically what it is happening, because non-cyclic photophosphorylation, when it will be excited, it needs to be come into the ground state, because it is excited. To cover up this particular electron transfer from photosynthetic electron acceptor center to plastoquinone, it is reduced. So it is oxidized. It is the electron is scarced here. So this particular electron has been obtained by a particular method, where H2O, water, basically this is the water, photolysis will be happening, water has been splitted down, it will produce 2 number of electron and 2 hydrogen molecule and 1 oxygen atom. This is a nascent oxygen. After 2 oxygen, when it will be added, 1 oxygen molecule will be released. So these 2 hydrogen will be utilized here, NADPH plus, nicotinamide adenine dinucleotide phosphate hydrogen atom. So cyclic phosphorylation is over and non-cyclic phosphorylation is over. You have to be just read, and carefully you have to be point out how the electron has been transferred from one location to another locations. So it is important for ATP production. Limited amount of energy ATP are produced in case of non-cyclic. So cyclic photophosphorylation is much more important in case of photosynthesis. Electron transport chains. So you have to be just remember the electron transport, where from it has been reduced, and whatever I talked into the particular drawing, it has been written. You have to be just read out some important points. You just have to be remember each and every question come from this section. So